In addition to, this significant body of data suggests that, in addition to B-cell mediated humoral immunity, antigen-specific T-cell response is also very important for effective immunity to influenza virus infection. Has been shown. Conversely, the multipotency of ASCs has been shown to be retained during long-term culture. The role of the VACV transmission cycle and sylvatic reservoirs remain a mystery. However, the role of small rodents in viral dissemination and as a link between wild and rural areas is becoming more and more apparent. We found that in this study, we found that high amounts of TBET induced ZEB2 mRNA expression and that ZEB2 was critical for the transcriptional programming of TE cell differentiation. Due to the This may be due to the fact that ovarian cancer has more modest ApoBE-C3b expression levels and mutation loads in comparison with some of these other cancers. Was used to. The t-test was used to compare continuous variables between treatment groups. Consistent with the These results confirm earlier conclusions that IL-12 is not essential for T-cell expansion in vivo and are consistent with the notion that IL-12 directly signals the downregulation of CXCR3 expression. In the absence of. However, neither the expression of SHH nor that of the PTCH2 GFP transgene was affected by this treatment, suggesting that in the absence of FGF signaling, HH activity is largely unaffected. in contrast to. We found a significant reduction of nearly half in pregnancy incidence among participants attending family planning visits, which is in contrast to findings in previous randomized trials. As well as it was inhibited in SNX9 and RAB9 KD cells, as well as in cells expressing GFP parkin.